Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Thanks. Well, thanks for coming, everyone. I do like these morning talks as opposed to our after-lunch departmental seminar, which is a hard one to, well, I won't say stay awake through, but it's just a bad time for a seminar right after lunch. I mentioned Turing in my title. Probably overreached myself a little bit there. Everyone say, "Oh, you're going to talk about Turing?" No, I'm not going to talk about Turing. I just put him up there because I wanted to talk about computation as well as ecology and evolution. I'm going to explain what this is a picture of a little bit in a minute. I want to start with acknowledgements just because I don't like doing them at the end because it just seems a silly place to do it. So I want to thank places that have supported me for the last 5 or 6 years, NJIT and Rutgers, Department of Mathematical Sciences. I came to NJIT, I was the only ecologist, and they didn't even have a Department of Biological Sciences. I came into the Department of Math. Now there is a Department of Biological Sciences. There's 4 of us, which is sort of a challenge, and many students, undergraduates. Funding has come from NSF, US Fish and Wildlife Service, State of Alaska. They still owe me some money, actually. And students that I have and collaborators over the years. This talk is set up to make an argument about where I think ecology can go in the future and where I would like to go, at least in the future. And the first part of the argument is that we have some huge gaps in ecological understanding that for a long time have been sort of ignored because they were hard. People have been working on them a long time. We still don't really know what the answer is. Why are some regions more diverse than others? Okay, this has been one of the outstanding problems in ecology. It's still not solved. There's dozens, if not, I won't say 100, but there's so many theories that explaining large-scale patterns in variation in species richness and other sort of things, latitudinal species gradient. Many theories, many plausible theories. There's not really a winner. They haven't all been brought together. How do species persist in fragmented habitats? Most habitats, well, many habitats are fragmented naturally. Many more are fragmented now that we have fragmented them. And so how species can possibly exist in these places, we haven't really got a handle on that. Fragmentation is going faster than we can understand it. Why some introduced species have become pests and others not. These are just some of the big open questions in ecology. And in many cases, I would argue that stems from large gaps in data that we simply don't have. Local influences on species distribution and abundance. There's lots of theories about that, but the truth is, for most groups of organisms, we have no idea what their local diversity is in most parts of the world, all their range distributions, for all the little things, okay, all the insects, probably 90% of phyla, we really have very little idea about their global distributions. We only really know the big things that we've been studying a long time, birds, mammals, flowering plants, those kinds of things. We have very little understanding of movement so far. This is an area which I think is ripe for a huge revolution in data collection. But up to now, it's basically been sort of brushed under the carpet. And I'm going to demonstrate sort of the way movement is incorporated in sort of classic ecological models right now in a ridiculously simplistic way because we don't have anything better. And what I'm going to argue is that developing technologies now are going to get us some of these data, and they're going to do it soon. And so we're going to have a huge flood of sort of completely novel information into many areas of ecology that we need to do something with. GPS, well, you probably recognize all these acronyms, so I'm not going to explain them. But I'll talk about a couple of them later on. So I think that we need to be taking advantage of advances in other disciplines and industries that are happening. One example I'm going to talk about now is the advances in graphics programming that's basically being driven by the gaming industry, which is what powers the advances in GPU and those kind of things. And I know I'm preaching to the choir here. Normally I'm not preaching to the choir when I say these things, but I am here, so I'm aware of that. But guiding them to be sort of a force for good, these great technological advances can be harnessed to do good things for the environment if we can just sort of show the way. And I think that there are many companies and things doing developments that if they only knew that what they are doing could be used, to really help the environment, they would be on board with that. But I don't know that we get that message out very well. And we need to be developing the tools to use the data when they start coming in. There's going to be terabytes and terabytes of information about, say, organismal movements around the planet. We need to know how to store those, how to analyze them, so we don't have a huge lag time in making use of them, and how to get that information back out into the ecological world where we can use it to test theories and develop new theories and really begin to understand what is happening in the environment. So I'm going to start off talking about movement. And I'm going to talk about, so these are the sort of two topics that I mentioned, movement and species distribution and abundance. And I hate animations, but I couldn't resist that one because it's the word movement. A movie just came out in the States called Light Water for Elephants. This is an elephant, and this is a huge barrel of supplemental water provided to it in Africa. When you provide water for elephants, this happens. Their populations grow exponentially, and even for elephants, that means that you get a lot of elephants in a surprisingly short time. It's a surprise to most people when it happens. And what happens then is they basically destroy the habitat that they're in. So elephants in parks, where there's no poaching, there's supplemental water, end up sort of destroying those parks. So there's an elephant management problem in southern Africa, which is that there's too many elephants in parks. One solution to that is to let them out of the parks again. In other words, sort of open the fences and let them go and sort of see what happens to them. There'll be semi-natural population regulation in that way. But we'd like to know, if we do that, where will they go? To do that, we looked at where they go when they're in the parks, and the contrast here is a little too much, but we looked at elephant movements from GPS-tagged elephants from 3 different parks, Etosha up there in Namibia, and Maputo and Tembe, which are adjacent parks here in Mozambique and southern Africa. And what we did with the elephants is say, what if elephants liked coffee? Well, okay, elephants don't like coffee, but someone who does like coffee is my wife. And she... If you want to know what kind of coffee she likes, you could look at where she buys coffee, right? And these are the 2 coffee places that are near us. You probably have Starbucks here, but you don't have Dunkin' Donuts, I think. You're lucky. If you looked at where she buys coffee, she buys about, when she's out and about, she'll buy it about equally from Dunkin' Donuts and Starbucks. And if you took the naive view, you would say that she didn't really have a preference between the 2 of them. But if you knew that in our local area where we live, we are surrounded by Dunkin' Donuts, and all the Starbucks are actually much further away, in the direction of New York City. I live in New Jersey. If you knew that, then you would conclude correctly that actually she prefers Starbucks because she drinks it more often than it is available in the local area. Or to put it another way, she invests more time going to find Starbucks than Dunkin' Donuts. So that cost, right, that travel cost, needs to be incorporated into understanding of habitat preferences when considering things like elephants. And although this has been done for quite a while when looking at sort of models aimed at sort of understanding human choices, it hasn't been applied very much to animal movements. So we did that. Seems kind of obvious, but it hadn't been done. So we did that. So the way we did it is we considered the elephant habitat selection as a dynamic problem. Where does an elephant go from every point where it is right now? So we have a series of GPS points, and for each time we model where the elephant is now and then how it chooses the next landscape location from all the available landscape locations. But we then weight them according to how far they are from where the elephant is now with an exponential drop-off function. So this is a habitat choice model, but it includes this distance from current location as a parameter, which is usually not included. As you can see with the coffee example, if you don't include that, you can get completely the wrong answer. So it's important to put it in there. Having done that, we were able to understand how elephants feel about water, which is that they like it, and how far they tend to go from it, which is about 2 kilometers. In the dry season and about 4 kilometers in the wet season. They typically don't go further than that.
Well, what are the curves showing us?
These are showing...
so I see distance from water to the horizontal axis, and what's the vertical axis?
Yeah, so these are all scaled. So what we have is we have a model with... These are actually fitted logistic curves, okay? And this is an exponential function, right? And then these are logistic functions for distance from water and proportion of closed woodland to allow them to have a sort of a threshold. And so what I'm showing, every line is a different elephant, and brown and blue are dry season and wet season. And what it is, it's the fitted function for that elephant's preference for that particular landscape feature. Okay, so I'm combining information from lots of elephants. So the solid lines are female elephants, for which we have the most data. Female elephants in the dry season, female elephants in the wet season. And this is the relative value they give to a patch based on its distance from water. Okay, so the best value is to be close to water, and this is how it drops off relative to that. They've all been scaled. The wax has been scaled so that they all start at the same point, just to make it easier to show them.
So can I think of it as kind of a probability, this? You know, a probability of 0.2 that it'll be further than 3 kilometres. Is it something like that?
It's not exactly a probability, but you can think of it like that, yes, essentially, yes. Yeah.
And the dotted line?
Those are for male elephants who are can be rogue in some cases. Male elephants will sometimes get thrown out of a herd and wander off by themselves, so their habitat preference is a bit difficult to understand because one thing they do is avoid. In many cases, they avoid other packs of elephants, so they're kept away in some cases from the good habitats. So male elephants, and they're also harder to catch and to put collars on, so there's also less data for male elephants as well. Well, we got quite a lot in Maputo. But they're less sensitive to water. Female elephants go in herds and there are baby elephants there, so they're very sensitive to the environment because of the juveniles that they have with them. So that's just an example of of doing habitat selection sort of as a movement model. And the result of that is once you have those models, you can go back to the landscape and create a sort of a quality map based on the underlying variables that you have and say, these are the areas that elephants will prefer the most, and then given their distance parameter, we can sort of smooth that a little bit to say, well, okay, well, this may not be the best patch, but it's near a bunch of good patches. So there's actually a river running down here, and there are some water pools around there, and so forth. So the contrast is a little bit high to see it very well. So we can go back and we can do that, and then potentially we can apply these models outside of the park to get an idea of where an elephant is likely to go. That's actually the next step that we've been talking about now. If elephants were to be released, where would they be likely to go? Here are some data that I've just started working on. Similar problem. These are brown bears from Alaska, from the Kenai Peninsula. So there are highlands, and then there are lowlands with lots of streams where salmon come, and the bears go down to fish. Some bears seem to more or less ignore the fish and spend their time moving around sort of in the uplands like this. That's a GPS track of one bear over 1 year. And here is a GPS track of another bear that is quite clearly going from sort of fishing hole to fishing hole. This is a bear that likes to fish. So we're trying to also model the habitat preference of those bears. And in particular, it's obvious that this bear likes water, but we're interested in when they're not at water, where are they, what kinds of habitats are they tending to frequent? The state of Alaska would like to know this because they want to minimize the number of times that edible hikers bump into hungry bears during the sort of the season when they're out and about and moving around. There's a challenge here because if an animal goes from waterhole to sort of waterhole, or fishing hole to fishing hole, you can't just use distance from fishing hole as a variable, because when they're leaving one fishing hole going to the next one, they spend half the time going away from it, which would tell you that they don't like water, and then half the time going towards it. And so you have to include, to do the bears, what I think we're going to have to do is include a time component. In other words, when a bear is at a particular location or at a fishing hole, over time, that fishing hole becomes less and less valuable, and therefore it's more likely to start moving towards the next one. So that's something that we're working on right now, because when we realize that bears were moving in this particular way. If we can understand movement better than we do right now, well, what can we do with it? And you might recognize this place. These are the islands off the coast of the UK that have sort of one of the longest ecological data sets in the world. Right, the sort of the breeding bird surveys from these islands go back over 100 years. They have almost no error due to the legions of fanatical bird watchers that occupy them every single summer and record every single bird that arrives. So they're one of the sort of the most accurate ecological data sets, have been worked on by many people for decades and decades. If we apply classic island biogeography to the data on those islands, so here we have the average number of species on an island and area and distance. And this is the classic sort of curve that says that the number of species increases with area in an asymptotic way. And apparently, there's no influence of distance whatsoever. It's expected that the number of species will decrease the more isolated the island is, but there's no evidence for that. And in fact, this model fits those data very, very poorly indeed.
But the distance from what?
Distance from the mainland, or from the nearest larger island.
Another island.
or another larger island. But for almost all these islands, it's from the mainland, because they're just dotted around the coast.
Okay.
These are usually the largest islands in their group, or they're somewhat isolated. So that's just species area. But in fact, for these islands, we have data on appearances and disappearances. Because we have such a long time series, we can actually look at colonization and extinction events, which are the processes that give rise to sort of average species richness. And what we noticed is that, actually, if I just go back to this one, what we see here is we see two islands that are very close to the mainland that have very low species richness. Here's one of them, not too far from here. This is Scalt Head on the north coast of Norfolk. And it's not that they don't have colonization, because that would be silly. They're so close to the mainland. It's that they have very high extinction rates, meaning that there are bird species that will breed on here for 1 year, and then they'll just go, and then they'll come back, and then they'll go, and then they'll come back. Their extinction rates are high. So there's an extinction rate distance function that is not in classical island biogeography at all. And what we hypothesize is that because they're so close to the mainland, because the travel cost is so low, that if conditions are less than ideal on this island compared to the mainland, that birds, they will just leave, that there's active dispersal. They'll just fly away. If it's a little rough out here, because it is kind of exposed to the North Sea, they'll just fly back here to the mainland. So that because birds are active, relatively intelligent mobile organisms, that they are using that, and they're making decisions to leave, that it's not a sort of classic extinction process, that it's just leaving, an active dispersal process. So in a sense, it's more like optimal foraging than island biogeography. So this is the classic model again. And so we said, well, for many data sets, we don't know anything other than species richness. We don't have this sort of long time series where we can get appearances and disappearances over time. But what if we took the classic model and just incorporated into it one more effect, which is dispersal for islands that are close to a mainland, that there is a sort of active dispersal function, that they can leave that island. And if you do that, you get this model. And I really like this model because it fits this data so fantastically well. This is the result of adding that extra dispersal that says that there is active leaving. And one thing you'll notice is that if you put that in, you actually then recover the expected long distance drop-off in species with isolation, which you expect from lower colonization from those higher islands. So it doesn't just give you what's going on here, it actually recovers the thing that was unexpectedly missing from the data in the first place.
How did you decide to make it sort of drop off so?
It's just a fitted.
So suddenly,
It's just one extra parameter that is a function of distance. There are two parameters now that are a function of distance.
Mm-hmm.
Okay, but one interacts through colonization and one interacts through extinction. So I can show you, if you want later, I can show you the equations for the model that go in there.
It's kind of a fairly abrupt one. You must have chosen.
It is. Well, it's active dispersal. It only really occurs over 1 or 2 kilometers that a bird will, because many of these are small body birds, a bird will just say, okay, I'm just going to leave. That it's no risk to leave. It has to be a very small distance. And we wouldn't expect it to operate at more than 1 or 2 kilometers, which is where it kicks in. So anyway, so we've actually, I won't show them here, but we've actually then sort of trawled the literature for a large set of species area distance data sets for mammals and herps, reptiles and amphibians and birds, and refit our model to them. These are things where they've already fit the standard species area distance models to them. We refit our model, and we find that for mammals and reptiles, which don't disperse over water particularly well, we generally don't find evidence of this extra thing. But of the 13 bird data sets that we looked at, 9 of them fit much better when you add in this little extra term. It's a much more significant model. None of them are quite as beautiful as the one I'm showing you here, which is the first one we did. But they are all statistically improved if you add in this sort of extra sort of active movement. Classic ecological models are just missing stuff because there's no movement. There's no real consideration of how organisms actually move. Most classic ecological models assume that organisms sort of drift around like pollen or seeds. All the dispersal functions are very, very simple and unrealistic for most organisms. Any bird? Does anybody know? Anybody can recognize these guys? No bird is here. They are black-crowned night heron chicks. And they are from Mill Rock, which is a tiny rock basically in the East River off Manhattan. And the species are actually nesting all over the sort of New York Harbor estuary area, along with a bunch of other herons, cormorants, and things like that. Since the Hudson cleaned up its act in about 1984, or we cleaned it up for it, they've been coming back. This is what the islands look like. This is actually the hospital where Typhoid Mary was incarcerated, which is on one of those islands. Now it's just a haven for birds. If you look at the numbers of breeding, or say nests, if you like, on the different islands over time, you get data that looks like this. So every line is a different island. And for many islands, there'll be no nesting for a few years, and then a huge spike, and then it'll stay colonized for a while, and then it'll drop right off again, and that colony will be absent then for many years. Meanwhile, other colonies come up and persist for a little while and then go down. If you look at that, you're going to say, well, it's not independent population dynamics. What's happening here is that the colonies are moving in year to year. They're just in different places. These are long-lived birds, and they reform their colonies every year. But the people who are interested in these, the sort of the wealthy Manhattanites who are members of New York City Audubon, are very concerned when this happens. And they get very upset about it, and they wring their hands, and they ask, well, what happened on this island? And they get very happy when this happens, and they say, well, we did something right. When I saw these data, I thought, I bet nobody's doing anything bad or good here. I think that probably that's just the way wading birds are. So what we did is made a very simple model, probably the simplest model you can make of wading bird colony formation, which is that every year when they come back from wherever it is they go down the coast, and we don't know exactly because they're mostly not tracked, when they come back, they have some site fidelity, but basically once a colony starts to form, birds will preferentially move to that one because they want to be together. They're colonial. And then that colony will build up until it's pretty much full. There is a carrying capacity for the colony. And then they'll start a new colony, and then that one will tend to fill up. That's that's all there is. There's no differences in site quality or anything like that. If you make that model, okay, you get data like this, and you get model runs that look like this. Okay, qualitatively, you get almost exactly the same kinds of dynamics, just from considering the way these birds move without anything else. So
your model said once a few of them land up there, then more join them, so they make a colony. You didn't say why they might then. Why might it die out? And once there's a lot of them there, a lot more will come until it's reached.
Well, it ends up being
it's
the stochasticity of the initial arrivals, like where they start forming the colony. So if a big colony was in island A in 1 year, it's more likely that if that was the biggest colony, it's more likely that the first birds back will be from that colony, but it's not 100%, right? They could be from another. The first ones back just go to wherever they were previously.
Oh, but it's all fresh every year.
It's all fresh, exactly.
It's
fresh every year, and it's slightly stochastic, right? There's a probability, and that's all you need.
I see.
It's all you need. And so you end up with this. So I go back to Audubon and say, look, don't spend all your time worrying about the smoking gun when the colony disappears. Don't ride off the island. It's not damaged. They'll probably come back in a few years. That's just the way wading birds are, right? Just trying to understand that sort of movement process. We move on to species richness and distribution. There's a lot of developments in this area. I was talking to Rob Goralnik at the University of Colorado about the sort of the map of life effort, which is an attempt to sort of combine all the information on species locations into a massive database, and then in real time allow you to sort of generate range maps using one of the sort of standard technologies, and to have a portal for doing all that. So here we see a particular, I can't actually even remember what species this is. This is one of their example species, showing that it's sort of locations of observations, and then this is some sort of estimated range, although clearly it's missing a couple of outlying observations there. But the problem is they want to do all of life, but for most species, these points are very scattered. We have very few collections of them. We control them from museum records, but we just don't know that much. So the first question I want to address is, how can we improve the number of these points? Because if we can have more of these points, then our range map gets to be much more accurate. If you didn't recognize the previous birds, you probably won't be able to tell what these are. So I'll just tell you, they are spider genitalia, female spider genitalia to be exact, from 3 different species. I know it's not the thing you wanted to see first thing in the morning, but there it is. Just so that there's no sex discrimination, these are the male equivalents. These are palps. I'm showing these because I want to talk about the spider system, which actually started off as a project by my wife, who's also an ecologist, who hooked up with a computer scientist at the University of Tennessee to try to see if they could come up with a system for automatically identifying those pictures, which are the diagnostic characters for spiders, to species. The idea being then, if you go out and you survey spiders, instead of spending 5 minutes per specimen trying to figure out what it is, or maybe half an hour per specimen trying to figure out what species it is, you can take a picture of it and have the computer, with some reasonable accuracy, give you an ID, or tell you that it doesn't know, in which case you spend half an hour doing it yourself. But if the computer can do the easy ones, then that saves a huge amount of time. And that way we can survey spiders and get a good idea of their distribution and abundance. For the males version, which is new, I've been working with her on this. So here we see some original images which are quite murky and are taken on a sort of a green screen background, although these are little plastic beads, so it's not uniform in some cases. So there's an image processing step, and contrast enhancement. It's over enhancement on the projector, but contrast enhancement there. We subtract the background. We then track features. These organisms, particularly the females, but also the males, they're just blobs. They don't have easy to characterize distinct shapes or features on them. They're just sort of blobby shapes. So we need a very generalized feature extractor. So going on from the original female version, we've been using Gabor wavelets, which look like this. And we lay them on this sort of multi-resolution. So there's like a huge wavelet that sits on top at 3 different rotations. And then we go down in sort of factors of 2 to finer resolution and generate a large number of coefficients based on whether or not there is that kind of texture at that particular place on the image, at that particular scale. And then identification is done by neural networks. And instead of having a massive neural network that identifies for the whole system where there are as many outputs as there are species, we have one neural network per species that is just trained to give a yes/no answer as to whether or not that is the correct identification. And it actually works. This is the success matrix, right? So the diagonal is successful identifications. The worst one is actually the very first one here. But we're getting 85 to 95% successful predictions for the males, okay, with a new system. And this is just in the last couple of months or so that we finished that up, which is similar to what they were getting for the females in the first place. So it's very promising. I mean, you can do this. And this is a hard test because this isn't all the spiders from one place, which are actually quite different from each other usually. This is all the spiders from a particular group all over Australasia. They're about as similar to each other as spiders can be because they're all from one family. So this is a hard test case. Actually, useful systems that are based on all the spiders in a particular place would be probably more successful. There is a website. I want to show you this one because I just before coming here, I broke it. But you can submit the images online. So yeah, I need help with my website. But you can submit the images online and it gives you the ID back again. It also gives you range map information that's pulled from a database as well, so you can double check the ID and do all those kind of things. I can show you there's an original version that goes with the females as well. Oh, the contrast here is going to be a problem. Oh well. The last thing I want to talk about in sort of ID systems that can help us get information is leaves. So I had a colleague who's in Malaysia now send me large numbers of scans of simple leaves from the forest that he works in. So this is a tropical simple leaf. It's not just a silhouette, I can assure you. There's all kinds of detail on it. So in order to try to identify leaves, especially simple leaves, where shape is something, but there's not a huge amount of shape information in a simple leaf because they are relatively simple shapes. The first thing that we do is trim off little spikes and any stem that's remaining there and sort of smooth the edge a little bit because the edges of leaves always have sort of environmental noise and things in them. And then we straighten the leaf up. You can't see it very well, but there's a midrib here. So in the previous image, what we did is we found the midrib using edge finding functions looking for horizontal edges. We then fit a polynomial to it, and then we just transform the leaf to stretch it out. Once we stretch it out, we can do all kinds of things with it, like look at its symmetry, and we can also look either side of the midrib and look at where the side ribs are, which is what we're going to do. Here's another example, and again, sorry about the excessive contrast. So there's the leaf, edges removed, straightened up. It didn't need much straightening in this particular case. In this case, you notice it's pointing the other way, but we can detect which way it's pointing by looking for the angles of the ribs. So we flip it so that all leaves are pointing the same way, so we know what is the base and what is the tip. So here's a few leaves that, that have been done and straightened and cropped. So we get 8 leaf shape features: the curvature, the aspect ratio, the roundness, vertical center of mass, mass asymmetry, the horizontal center of mass, the difference in the horizontal center of mass of each side. In other words, some leaves are fat on one side they're fat at the base, another leaf, on the other side they're fat at the tip, right? So there's a sort of horizontal asymmetry. And we can look for sort of small scale serrations around the edge, anything that gets past our filter as a sort of just a measure of sort of toothiness. And then we look at the side ribs, because we're bringing by, we know that what's interesting about leaves is their structure, and the ribs are part of the structure. And so we go in looking specifically for that. So we take 2 slices around the midrib. And what we do, and so there's another slice up there, and you probably can't see it very well here, but you'll see better in a minute, right? That these have the side ribs running across them. And we convert to grayscale, that's a little better, sort of an enhanced contrast. And then we apply a set of custom rib finding filters, which are just essentially edge filters, but they're quite large and they're at different angles, so they pick up particularly angled straight lines. Run a set of filters across that, and you get a series of filtered images like this. If we binarize them, we get that. We add them together, we get this. Inverting them, expanding them to make sure that any broken ones get joined together. Remove any that are below a size threshold. Thin them again, and you can't see it here, but there are very thin white lines running there. And then finally we find their locations. So now we have a series of locations of the ribs, and this one actually at the end, there's another point there as well. So we found where the ribs are. And here's another example, the original leaf that you can't really see, and the ribs detection here. And then what we do is we say, well, for some leaves the ribs get further apart as you go to the tip. For some leaves they get further apart and get closer together again. That's really about as complicated as it gets. So we fit a function to interval to next rib against rib location, and this is a quadratic polynomial fit, actually to the log transformed intervals. And here we also detect the slope by fitting a line to each rib one at a time. We also have a function rib slope as a function of rib location. That typically increases or decreases monotonically, usually increases. And so from these fits we extract information. So we end up with 8 leaf rib features: mean rib interval, fraction of leaf length, interval change, final interval change, the error, how noisy are the ribs, and whether they're symmetric on either side of the leaf. Some leaves have very symmetric ribs and some don't. So at the end of all this, we have 16 leaf shape parameters and rib shape parameters that we can apply to simple leaves. Does it work? I'd love to tell you, but I see only got as far as this. Right? We're about to go and now look at the discrimination ability using these features. What I want to point out here specifically is that all these systems sort of use known biological information about the specimens to focus in on particular things to look at. In other words, we take our taxonomic and systematic knowledge and apply that to the system. There are quite a few people also trying to do species ID stuff, often purely in departments of computer science. And what I notice is that they tend to they'll throw the latest algorithm at a problem and see what happens without really thinking or even perhaps knowing anything about the biology of the organism in question and what a systematist would look at as being diagnostic of those things. So, for example, there is a leaf detection algorithm that's been applied to US leaves that just looks at, there's just one kind of function that looks at distances around the edge of the leaf. And they've published it as a great success, but I looked at the results. It's not a great success at all, actually. It doesn't work very well. And so I think that we can do better if we take biological and ecological knowledge and apply that to these algorithms. And the combination of the two things is where the power arrives. How am I doing for time?
You've got about 5-7 minutes.
Okay, I shall go very fast then. this is the typical range that you would see for a species in a guidebook, right? It's a blob. This is also the area of this blob is the kind of range that the IUCN uses when assessing extinction threat, right? So the extent of occurrence. But in reality, if you look at where the forest that species inhabits was, or could potentially be, it's actually a lot less than that. And if you look at the range of elevations that the species is being found at, it's often quite smaller as well. And if you cut those three things down together, you end up with an expected range that looks like this, which is a lot different and a lot smaller than the original range. If you go ahead from the original wet forest and you look at where forest is still remaining, there's a lot less of that also. And so if you cut the range down further, you end up with an estimated potential habitat, a potential range I should call it, for a species that is often 100 times smaller than the extent of occurrence, or 10 to 100 times smaller than the extent of occurrence, and extremely fragmented. If we understand species richness and distribution, so this is something you can do with just readily available data. Satellite imagery, elevation layers are all just stuff that you can download and do this with almost anywhere in the world. But if we can improve sort of our understanding there, what can we do with it? This is a spatially explicit metapopulation model that models the persistence of species in small patches as a function of their extinction in those patches and whether they receive colonists from other patches. Well-known model has been around for many years now. It has a dispersal function in there that I'm deliberately leaving undefined. One thing we've done is there's a metric called the metapopulation capacity that you can apply to a range that requires only that you know areas and interpatch distances or movement rates. And that summarizes the ability of that landscape to hold a particular species, which we can go and apply to those ranges that we just made by using our satellite imagery and using our elevation maps to ask what really is the capacity of that species' potential range to hold that species. And we can use that to come up with a different way of ranking species in terms of their threat based on fragmentation. So this shows the species, this is the current IUCN threats for a set of species actually in the Atlantic forest of Brazil. So this is them ranked based purely looking on remaining range. So this is what happens if we improve the sort of the blobby extent of occurrence to just the expected remaining area of habitat. And you'll see that the rankings are often very different. Things that are listed as being vulnerable or only vulnerable, which is the lowest level of threat from IUCN. In this case, this species has the fourth least remaining range of all the species in the list and probably should be ranked a lot higher once you do that cut down. And here's what happens if you rank according to metapopulation capacity, which includes not just the remaining area, but also the fragmentation and the way that influences the species' persistence in those sites. You'll see that there's potentially a very different ranking of species from those two things. So we're in the process of writing up this as an alternative metric for potential use for ranking species for IUCN that you can apply to almost any species because it uses readily available data. You can also value individual forest patches according to their contribution to the ranges of all the species that live in that area. This is Central America, the Guatemalan highlands, and the patches here have been colored according to the value of the individual pixel to the range of all the species that overlap there. So now we can explicitly rank. Here's a sort of a 3D version. The only remaining forest is in the tops of the mountain ridges. All the rest is gone. But this is the highest area. Here is a sort of a plateau region here that's probably the furthest from development. And many species ranges overlap there. And here's the same thing. This is now the coast of Brazil. Rio de Janeiro is there. And so this is Atlantic forest, probably the biggest remaining patch of Atlantic forest. So that's another way. So we can value those. We can also do restoration. Once we have this model in place, we can say, well, what happens if we put back individual pixels that were forest originally and are not now? And we can rank those according to how much they improve the ability of the landscape to support the set of species that's on there. And we can test out corridors and individual pixels and actually say, okay, if we can just buy 1 square kilometer of forest, which one is it? Oh, it's this one right here, which is a very powerful tool. Putting it together. So in these models, right, again, talking about movement and bringing that back to how organisms persist on landscape and diversity, this is what usually goes in there for dispersal. This is just an exponential decay function that assumes that individuals move in straight lines between patches and just have some constant probability of dropping out of the sky as they go along. This is a slightly improved version that we're using right now, which is based on dispersal in fragmented forests. It's just a slightly more heavy-tailed distribution that says that birds can go long distances if they want to, right? Which this doesn't really allow. Okay, this drops off to essentially nothing over more than a few kilometers. But can we do better than that? So here's what we're trying. So here we see, what you can't see is in the bottom of these holes are habitat patches, right? So this is actually a map. And one habitat patch is raised up and the rest are in holes. And the surface is generated as a diffusion map, assuming that there is some chemical diffusing out of here and going and being absorbed here. And so we create, and the whole point of doing that is just to create a landscape that has sort of basins of attraction around each patch. And the bigger patches have deeper holes, right? They're set lower down. So they have bigger basins of attraction. And then what we do is we take organisms that are leaving one patch and we have them just walk down gradient until they fall into a hole and find their way to another patch. It's a very simple, it's not really an ecologically mechanistic model, although it would be if the organisms were chemotactic. But it's just a way of coming up with something different than e to the minus alpha dij, which is what gets plugged in right now. So here's sort of how it works. So now we have organisms heading out from all around this particular patch, and this shows where they end up. Birds do fly like this. They don't just fly in straight lines ignoring what's there and bump randomly into patches. They fly to places. So we do that, and we can then expand that out to the whole landscape. And from all these patches, we can generate a new matrix of relative dispersals between patches that's different from simply looking at some simple function of the distance between them. We can also include distance because some of these are still long distances and we can still reduce it if that's a dangerous journey. But it's much more like the way organisms actually move. And if you don't believe me, I had some undergraduates follow over here woodlice, pillbugs, roly-polies, whatever, many names they have, on some artificial landscapes where we track them with video cameras to see where they went. And yes, of course, when they leave this patch, most of them go here and not here, even though the distance is the same because there's just a lot more edge. Right? So our new model works better in this experiment. It was a very simple experiment. They had a good time. We've been pushing this out to New York City. Okay, so I have the vegetation patches of New York City and all the buildings at 3-meter resolution. Okay, I'll zoom in a little bit to show you what it looks like. This was rasterized for me. It's a very, very low, it's about 15,000 by 15,000 image, or matrix if you like, showing the heights of all the buildings in New York City. This is part of an NSF-funded project to see how the connectivity of habitat patches is affected by the buildings. What I started off doing, and this is very murky, so I'll just describe it, is I started off having individuals move, sort of doing discrete dispersal, pixel by pixel, out of Central Park. This is a corner of Central Park now. And this is Morningside Park, and this is Columbia University. Going pixel by pixel, it's incredibly slow, and they get stuck in alleyways and blind alleys, and it takes them forever to get anywhere, and I have to do this for the whole city, so it doesn't really work. So then, I did it for a while, right? And these are organisms who end up in Riverside Park when they leave Central Park, which is over here. But these guys got stuck, and they never come out of there. Some guys got stuck here. It's a little bit unrealistic, and it's incredibly slow. So I turned to image processing. I took the building map and the vegetation map and did a watershed transform, which creates all these lovely paths down the middles of all the roads that don't go into all the little blind alleys. Having done that, I then found all of the intersections and walked from each intersection to the next intersection and then just created a single edge in a graph for that, saying the organisms just hop from intersection to intersection. So I end up with a graph of New York City, which looks like this. This is Central Park, right, obviously. With all the edge around it, and then these are other parks, Riverside Park. So this is just a sort of graph representation of it. Once you have this, you can then have organisms walk from vegetation patches to vegetation patches along this graph, which is about 1,000 times faster than having them walk on the actual landscape. But it yields essentially the same results very, very quickly. So now we have a graph where each of the points is a vegetation patch, and these are the connections that organisms make between those vegetation patches. That's a graph representation of it. But when we blow it up to the city, we get this. And again, the colors are not fantastic, but we have this dispersal matrix between vegetation patches that we can now plug back in to these metapopulation models and understand the persistence. And we can do this again. This is for all buildings, so for organisms that can only walk along the ground, any building is a barrier. Or we can say, okay, let's only take buildings that are over 1 story or something like that. which eliminates a bunch of buildings down here, and we get a different looking matrix. I don't have an image of the matrix, but these are the movement patterns. And so we can actually look, and we actually have historical data sets on building height, so we can see how the change in building over time has affected the ability of organisms to persist in the vegetation patches around the landscape.
Do you have data on the buildings? Do you have data on the creatures?
We do have data on...
Is there any reason to suppose this is connected to reality?
There is. We have data on a number of organisms. We have quite a lot of data on birds, of course, because there's always lots of data on birds. And we have a few data on things like, we have some data on mammals, we have some data on dragonflies. It's not systematically collected, unfortunately. And that's the big problem, because movement data is one of the big gaps, so we just don't have it to really test ideas like this. This is much more plausible than e to the minus alpha dij, but we don't have the data to really prove it in most cases. Which is why I end up talking about ecology and technology, in which I mean a combination of sort of hardware and computation, ways to do this. I've been talking about ways of sort of censusing animals better. If we can census them, we have a better idea of the distribution and abundance. Movement is something we would like to. I'm just going to finish off by talking about my NSF project to make an automated underwater system for reef fish. And here it is in a testing tank. We have two cameras looking into a volume with two sort of black panels as background. Here it is in the New York Aquarium at Coney Island, which has an exhibit that is based around a Caribbean coral reef with quite a lot of fish in it. So it's sort of sitting there right in the middle of the tank. And here in absurdly high contrast is some video from... So we have two feeds, right? Because there are two cameras looking at right angles. There's one example. There's a Spanish hogfish. There's another hogfish. This is a big ugly guy. Getting a little too close. Nice teeth. And then here are some, these are French angel fish, which are very hard to detect because their background is they're exactly the same color as our background panel, except for the yellow spots. Swimming around in there. So this is with a collaborator at Rutgers University, but we extract the fish from each of the camera feeds. We track them. We then link the image from one camera to the image of another camera to do pose correction and scaling, because we get some idea of how far back. Because we have cameras at right angles, we know roughly where they are in the three-dimensional volume and how big they are. Feature extraction and recognition. We just started doing this part, and it's very simple. We have eigenfish components for the outline of the fish. And it says color and texture parameters. We're actually just using color right now, because you have good color information on coral reef fish, classification by simple nearest neighbor, weighting each species depending on whether shape is more important for that species or color. And these are our success rates. We only just got this result a week ago after finishing the experiment. And this is how successful we are identifying the 8 most common species that are in that system. And so for most species, we're in the 90s, 95 or above. This is the trickiest one. This is actually our first cut at it. We have some more features to extract that we think we're going to improve. And we have some more species to add too, but the image processing steps need to be improved a little bit as well. But this also does work. You can do this. In the future, I'd like to do it using 3D model matching. So we don't have to have 2 cameras and a huge system that's this big, but we can have a smaller system with 1 camera, because we can take a 3D model of a fish, right, and we can spin it around almost every conceivable orientation in a second in a GPU and try to find a match against our single 2D image from our camera feed. I think that's going to be a very, very useful system, and that's something I'm trying to get involved in right now. But I've been doing it with volunteers, and it's very difficult because they keep going away and joining game companies before they finish helping me, sometimes before even graduating. But that would be a really cool thing to be able to have, and even to have for sort of public feeds from the ocean as a fish swims across. You can imagine the ID just popping up right there. This is what it is. And then information about its range and its endangerment status and things like that. And when we do all these things, it's very important, I think, to get them back out into the real world. So this is a website I designed. I first, a very long time ago, although I've redesigned it a couple of times since then, that has tools for doing ecology, common things that you need to do for ecological analyses. Graduate students seem to do these things a lot, it turns out, because they're always emailing me. But for example, estimating species richness, there is a tool out there that does that, Estimate S. But this is an example where you go to this tool and you can send your own data file, which is a simple matrix of species abundances. And you can also run one of the test data sets. This is a classic data set that's in all the publications about species richness estimation. But you said you can submit your own data matrix and you cross your fingers. I always cross my fingers because... and you get a whole lot of information back. Information about your data set, all the relevant characteristics. You get estimates of the true species richness using a variety of different metrics. And you get back these... where are the rest of the pictures? Are they coming? Yep. Oh, here they come. All right, very slowly. You get back... you get back estimates. This is an estimate of the quality of the estimator. You want it to have asymptoted before the end for it to be at all reliable. And these are all the standard things. Like you can go to a publication and see exactly this stuff. But now you can do it, right? And you can just send your data set from anywhere in the world and get the results back. You don't need to download any software or worry about compatibility or anything like that. And you can... This is the rarefaction curve, sample-based rarefaction curve. And here's all the data if you want to get it. And you can also click on a link and just download that if you want to make your own plot and get it as a text file. And so, there are other things in there. I won't demonstrate all of them. But the vision that I have for this is that when someone does a new technique that's a real advance in the field, it should go on here at the same time the paper comes out. Instead of it being published, nobody reading it, somebody eventually reading it, and then asking the person who wrote it how to do it 5 years later, by which time people have moved on, that there could be a sort of like a tool bank where when you publish a new thing, that it should sort of immediately just be available as the latest technique, the latest improvement in the measurements. So one thing we're going to do with Lucas Joppa, who's here... Oh, I'm sorry, I've got to go back to my laptop. Is he and I have been working together on this new method for estimating the total number of species to be discovered, which yields plots like this. And in the next paper that we're working on, we're going to put this up on Ecotool simultaneously with the paper being published, so that people can immediately do it, as well as reading about it. Anyway, I'll stop there. I think I just came in under the wire. Thank you very much for listening.
Well, obviously, people should feel free to go, but also if anyone just wants to hang around and ask a couple of questions, then ask away.
Yes.
So, I'm not an expert in this area, but from listening to your talk, it sounds as if it's biologically sophisticated. Right? You stress that it's important to understand the biology in order to not do stupid computation, but computationally fairly straightforward. So it's kind of applying routine methods that are available in machine learning.
It's stuff I've been able to do myself, so it can't be that clever.
Is that your goal? So to take, as it were, off-the-shelf chunks of computation and apply them in an intelligent way in this area? And I guess I also wondered, going back to the beginning of your talk, where you said, "Oh, it's a flood of new data going to be available. We're about to make big steps forward." That I didn't. Did that come out in your talk?
No. So I'm thinking of examples like there is a move coming out of the Max Planck Institute to put up a dedicated animal tracking satellite that will be able, for instance, to track small things rather than just big things like elephants, which wear these huge collars. That you'll be able to track things down to sort of with 5 gram and 1 gram collars. So if that comes to fruition, right, it's called the Icarus project, right? If that comes to fruition, it's going to suddenly yield a vast trove of tracking information for small organisms that we just have no idea how they move around at all. So that's the kind of thing I'm thinking about. Advances in RFID technology and allow you to put tags on much smaller organisms than you could do in the past. Battery improvements. So I'm not working in those areas, although occasionally I email people who are and say, how is RFID doing? Can I put it on a beetle yet? This sort of question. The answer is soon, but not quite yet. But it's things are getting smaller and more power efficient and to do those kind of things. So they're coming along, and once we have that stuff, the sort of the models that I've showed, which are very ecologically plausible. If you show them to an ecologist, they will say, "Oh, that actually does look like the way birds move," but we can't prove it. It just looks plausible. It works with our intuition, but soon we will be able to test them. So what I'm saying is we can sort of make those models, get them ready to be tested, and then help sort of guide as the data comes in, the acquisition of the data to test those models, and then very quickly we'll have a better idea about how these things are happening. I'm not sure. Did that answer your question?
Can you give an example of an insight that you got from applying computational science that you wouldn't have got if you'd just been a biologist? Maybe that's the kind of thing I'm after.
Well, yes, so I think one example would, it probably comes down to the movement thing, which started off not with computation, but just with an analysis, that's more like a statistical analysis, looking at the island birds and finding this remarkable thing where the extinction rate was this very strong function of distance that's a completely not expected result and is not published anywhere, and trying to figure out what that was and how that could really work. That led me then to thinking about, it has to be that movement is not the way we usually model it, right? And so then that's when I started putting together sort of the alternative ways of looking at movement, like so diffusion models or diffusion following a gradient, those kinds of things, which are not incredibly new ideas, but had not been applied in this context before. And when I saw the kind of results that they produce, right, the kind of predictions that they make, that they do fit more with the data that we have. And so to me that sort of confirmed that our hypothesis about, based on that sort of one data set, that really it was active movement that was the important key, does actually seem to be plausible. There's still work to do. For the stuff based on the forest fragmentation, we have data on the presence or absence of species from many of those patches. Okay, so we're just generating the predictions now about whether the species expect to be there or not, and we can generate those predictions based on the original movement model or on the new movement model and test those against, not against actual movement data, which we don't have, but against whether the species are there or not, which we do have some data on, not as much as we would like, but we do have some data on that. So the one can go ahead and make predictions that go some way towards testing the things, but what we really need is actual movement data, but that's a proxy for it.
The high rate of extinction of birds on islands close to Britain, at least, could be down to the presence of, say, rats.
Yes, it can.
Extinction of, failure of nesting.
It can be.
Is it possible to distinguish?
So these data come from bird observatory reports, right, which usually end up at Oxford in the Edward Grey Library, or you can get them from the things themselves. And those reports, although the data get pulled out of those, and put in a sort of a database, they do record what is happening on the islands also in each year. And so whether there are some of the islands have people on them and some don't, right? Some have sheep and some don't. That information is there. Whether there are rats on the island or not, that stuff is actually usually recorded, particularly if anything new happens. The reasons for nest failure are usually recorded as well. They know where every nest is on these islands, and they usually say if a nest fails, they say, oh, it was, the eggs were eaten by something. So those data are there in the reports, and they don't correlate strongly as you might think with the distance. It's not that those two close islands have, tons of predators and things that the distant islands don't have.
So the low species richness can't be explained entirely by
right. In fact, not really very much at all. I mean, we've thrown in the analyses that we've done, which I only showed like one figure from, but we throw in a bunch of other parameters that we have as well. Habitat diversity is different on the islands, and we have different measures of that. We put that in. No matter what else you put in, this effect remains very strong. Actually, it's the strongest one in the data. It's actually stronger than the area extinction effect, or roughly equivalent to it. So yeah, we did pass it various ways, and there's a paper that sort of talks about it in some more detail.
I think we probably ought to leave it there so we can thank Gareth once again.
Thanks for listening.